If you imagine a big meal of protein, it contains a lot of steak, it contains some beans, some pulses, generally high protein containing things. All these things will contain long chains of amino acids, which are the proteins, the polypeptides. When you eat these, it will be broken down and digested in things like the stomach, the small intestine, before it is finally in its free amino acid form, and it can be put into the bloodstream, absorbed into the bloodstream. These hydrolysis reactions will be catalyzed by hydrolase enzymes. This means they utilize water to break down the links between the individual amino acids so that they can become separate. That's how that specifically works anyway. But once you have these amino acids inside of your bloodstream, if you imagine all the energy releasing processes that occur inside of your body, the glycolysis, the fatty acid beta oxidations, you've got the Krebs cycles, you've got the oxidative phosphorylations, all of this stuff utilizes energy, and none of it contains a single nitrogen. Proteins, amino acids, they all have nitrogens. It's part of their chemical structure. An amino acid, the amino part is representative of an amine group. The acid part is representative of a carboxylic acid group. This means that somehow the nitrogen has to go for it to be involved in any sort of metabolic process that actually involves energy. Sometimes these amino acids can be involved in things that need nitrogens, because nitrogens are a very useful thing inside of the body. They can be used to produce nitrogenous bases, which are useful for DNA. But the thing is that these energy containing processes, these energy releasing processes, they don't have any nitrogens, and so somehow the body must get rid of them. Problem is that it can be a very toxic process to deaminate these proteins, these amino acids, while in some cases, some amino acids, while they've got that main little amino acid structure, some amino acids will have extra nitrogen groups on its sort of molecule. Glutamine and glutamate are the big ones of these. Glutamine, you have the sort of amino acid group, then you have the another like amine group at the end. Glutamate, it has the amino acid group, but then it will replace the amine with an oxygen double bond. This is important because in some cases, when some proteins need to be deaminated, it will use either an alpha-ketoglutarate, which is where the initial nitrogen is also gone from the amino acid group, and turn it into glutamate, where that oxygen ketone group will then be converted into an amine group. Alpha-ketoglutarate and glutamate are not infinite things. Alpha-ketoglutarate needs to go round the Krebs cycle. It's an intermediate in the Krebs cycle, so it can't always be used to deaminate proteins because it needs to produce energy inside of these cells. So what happens to the nitrogens if there's not enough of this specific process to get rid of that amine group? Ammonia is the answer. Straight up ammonia, a very toxic substance inside of the body. Can you imagine that was the answer that I was going to give you? That's the way that the nitrogen group is broken off. If protein needs to be deaminated and there's no alpha-ketoglutarate available, it's going to come out as ammonia. Straight up ammonia, NH3. Very poisonous. And so if you don't have the enzymes that are needed, if you don't have enzymes in the cycle that I'm going to talk about in a bit, you can develop hyperammonemia, which means that you have a toxic buildup of ammonia inside of your bloodstream, inside of your cells, which isn't good news whatsoever. How does your body deal with this ammonia exactly? Now, ammonia is safely converted to urea during a surprisingly long metabolic process. You compare the two molecules, don't seem all that different. It's like you've got two ammonias that have had one hydrogen taken off them to be attached to a carbon, which has an oxygen double bond. Doesn't seem all that complicated. Thing is, it's very complicated, and I'm going to go through this urea cycle in the preceding minutes. First of all, CO2 that is present inside of the bloodstream, you have just sort of released it into the bloodstream from the cells that have gone through the Krebs cycle, which releases carbon dioxide. This will be dissolved into a solution, and it will react with the water that is the majority of the solution itself. This will form bicarbonate using the enzyme carbon anhydride. Now, the bicarbonate atom will disassociate one proton. This bicarbonate carbonic acid, a very weak acid due to a few things that I'm not going to explain, but it will disassociate one proton to make an HCO3 minus ion. This is then goes back into cells to start the urea cycle. And in these first three steps that I'm going to talk about, It's all catalyzed by one single enzyme. The first of these 3 steps is the HCO3- ion being added to adenosine triphosphate, and this will then turn into adenosine diphosphate, as well as an HCO3 PO3 2- molecule. I'm not entirely sure what this molecule is named. I'm pretty sure it's just going to be carbonate phosphate or something in that realm. But then you take that sort of carbonate phosphate, whatever you want to call it, you add it to an ammonia, you add it to the NH3, as well as a hydroxyl group. This will turn into a weird sort of part of phosphoric acid. It's where one of the sort of OHs that will dissociate is still got a hydrogen atom, but the other two just don't, and this will mean it has a 2- charge. This will also form water, as well as NH2COO-. Can't remember what that is called. Probably something, honestly, it's completely escaped me what that's called, but I will put it up on the screen. And then the third step, the final step of this single enzymatic reaction, is you take that NH2COO-, add it to another ATP. This will turn into carbamoyl phosphate, which has the formula CH2NO5P2-, as well as an ADP molecule. These first 3 reactions occur in the mitochondria, as well as are catalyzed by the enzyme carbamoyl synthetase. And the fourth step is you take that carbamoyl phosphate, you add it to a ornithine molecule. This will end up turning to citrulline. This reaction is catalyzed by the enzyme ornithine transcarb amylase, as well as the fact that this ornithine transcarb amylase needs some zinc and some biotin to function. Zinc helps to stabilize the enzyme, means that it just doesn't sort of, things just don't like happen to it. Some enzymes can do a lot of weird things, phosphorylation, etc., etc. It helps to just stabilize the enzyme, could be involved in the production as well. Biotin is a coenzyme, it's a cofactor inside of the production of citrulline inside of this urea cycle. This reaction also occurs in the mitochondria, and it's the last one that does occur there. After this, citrulline will be taken out of the mitochondria, probably by some kind of protein-carrying enzyme, or it will just diffuse or osmose. Then citrulline will be added to aspartate, as well as an ATP molecule. This will turn into adeninosuccinate plus an AMP, adenosine monophosphate, as well as a pyrophosphate. Now, the ATP to AMP plus a pyrophosphate, not really going to explain that one. I assume it's something to do with some kind of energy levels, but that's not really my area of expertise. This reaction is catalyzed by the enzyme argininosuccinate synthase. You take that argininosuccinate and you react it with the enzyme argininosuccinate lyase. This will turn into fumarate as well as arginine. And so fumarate, as a waste product of this reaction, will probably then go into that cycle as well. After this, you take that arginine, you add it to an H2O molecule. This will turn into ornithine and urea. This reaction is catalyzed by the enzyme arginase, and all of these reactions that have happened ever since we moved out of the mitochondria all occur inside of the cytosol, cytoplasm, whatever you want to call it, and that is it. You've got urea. Urea will then, it's a very toxic thing, less toxic than urea, it has to be said. It will osmose out of the cell into the bloodstream. The kidneys will then get rid of it, put it into the bladder, and you will then excrete it when you go to the toilet. That is how urea is dealt with inside of the body. Not urea, that is how ammonia, while urea has to be dealt with as well, that is how ammonia is dealt with inside of the body. Not good news at all. Bad thing, but your body is amazing, and the human anatomy is a pretty incredible thing, and of course it will find a way to deal with the majority of things, unless you just give it too much of a hard time, like the majority of human beings also do on the planet. And with that, that is it for how the urea cycle deals with the ammonia from these excess amino acids. Not really excess amino acids, because they will then be metabolized into certain things. But if alpha-ketoglutarate is not available to turn into glutamate, then this process is used. And that is an incredible thing that your body does. But I hope you enjoyed, and I recommend subscribing, because I love this community of young tribal people who are willing to improve themselves so much more than they already have. And good luck.